Our first speaker in this session is Cenk from University of Indiana. He's a very multidimensional person. His studies and papers in algorithms theory, computational biology, bioinformatics. He's going to be talking about the algorithmic approaches for tumor phylogeny detection.
Thank you.
Using sequencing. Please.
Thanks for organizing the workshop. It's been fabulous. I had to say it. So we'll be talking about combinatorial optimization strategies for tumor evolution modeling. Many of you have probably seen this or similar pictures. According to the clonal theory of cancer evolution, from the emergence of the first cancer cell on the left, maybe I do need the pointer. Thank you. To the time of diagnosis, tumor cells acquire mutations, some of which may give them proliferative advantage compared to the neighboring cells, and they may eventually come to dominate the tumor tissue. And given a mixture of these cells, especially in a bulk sequencing experiment, the goal you may have may be to identify the number of distinct cell populations defined with respect to the mutational composition, the prevalence of each subpopulation, and tumor purity, the specific, obviously, mutational composition of each one of these populations, and finally, the evolutionary tree for the tumor. A cartoon picture depicting this process is here. From the healthy cells, assuming that a single cancer cell with the first mutation, you can see how eventually the tumor cells acquire mutations, and the prevalence would be depicted as the length of the line segment here. So in a typical study, you're given the signals as a mixture, and you'd like to deconvolve them, trying to figure out, obviously, the composition of each one of the cells. And the eventual output would look like this. So this is a clonal tree with distinct subclones here with associated prevalences. So obviously, bulk sequencing provides you the necessary data from a mixture of cells, and mutation calling gives you what specific mutations each one of these cells may harbor in bulk. So you'd like to deconvolve this. How do we do that in a single experiment? That has been a major challenge. And as you can imagine, there are multiple solutions to this. First, we have to formulate this as a combinatorial problem, then see what exactly we can do. The input to this kind of a problem would be given as a table where each mutation would be associated with the number of reads supporting it and the number of reads that do not harbor it. And you should also have an idea about the copy number. Ideally, you'd like to focus on copy number neutral regions, provided that you can be sure of this. Maybe for the sake of this talk, we can focus on those with 2 copies exactly in the diploid human genome, but you can see how it generalizes to multi-copy genes too. And this doesn't necessarily have to be coding. It can be done on non-coding parts of the genome as well. And based on this, you can compute the variant allele frequency that many of you may be familiar with. And the variant allele frequency would impose an order, at least a partial order, on some of these mutations in provided that, first of all, they live on the same path from root to one of these leaves or multiple leaves, and provided that your measurements are reasonably accurate. There are multiple tools for figuring this out from one or multiple samples obtained from the same individual. Two of them happen to be from my lab, in particular SITAP here, which focuses on multiple samples obtained from a single patient. It could be spatially distinct or temporally distinct. Or CTP single focuses on a single sample with relatively low coverage, which is necessitated by the reality that we face in a clinical setting, where obviously the cost is a big issue. And each one of these tools eventually aim to come up with a tumor phylogeny. So in its simplest form, if you have the typical 30-40x coverage data with short reads, how do you figure out the clonal composition of the tumor? The idea is very, very simple. You're given, say, the variant allele frequency depicted in the vertical axis, and the total number of reads that are with that particular variant allele frequency depicted in the horizontal axis. And you can try to see that envisioning this as a random process with respect to coverage, and envisioning that there are a fixed number of small number of distinct clones, you can try to cluster them together and not only infer the number of clones, but which of these mutations belong to which of these clones. So eventually you'd like to perform a clustering based on the distribution under the assumption of normal distribution. So this is relatively simple, and there's a long process coming up with these clusters. I'm not going to get into the details as much, but obviously there is a clustering ambiguity. There could be subclones with similar cellular prevalence, and yet they would be aggregated into a single clone, and ideally we'd like to be able to distinguish one from the other as well. So according to a particular distribution like this, say two mutations are clustered together, the true tree would be quite different from the inferred solution based on the clustering. In fact, there could be many, many solutions, and there's no way to truly distinguish one from the other, especially since the linear chain topology is always going to be among the permissible topologies. So since this is a trivial solution, many of the tools would simply come up with a simple linear topology. In reality, you're trying to figure out whether there are distinct subclones in the tumor sample. So as I said, our first tool, CTP single, aims to do this clustering. The next tool that we developed called SITAP, in fact, from a temporal point of view, SITAP came first. You can have multiple samples from the same individual with a different mixture, but eventually supporting the same topology. Now you're trying to come up with a single topology that is a permissible topology in each one of these samples. So you can think that each mixture provides you a bunch of constraints on the topologies, and your goal may be to find out the topology that satisfies all. And I'm not going to get into the details of the objective or all the constraints. This is basically a QIP, quadratic integer programming formulation, where the objective is minimization of measured variant allele frequencies and the estimated variant allele frequencies per clone that each mutation is assigned to. So it's not a very difficult problem. The only thing that you have to know as a prior knowledge is the number of clones. You can obviously generalize it and assign each mutation to a distinct node in the topology, which would automatically minimize the objective. It will make it 0. However, that is not the ultimate goal. So you can come up with Bayesian information criteria or other criteria to decide on the number of clones, number of distinct nodes in the tree. So many of you may know that it's very difficult to obtain multiple distinct samples from a single tumor. Is there a way to add new signals on a single solid tumor biopsy? And liquid biopsies provide that opportunity, and we have been working with them. As some of you who may be also dealing with liquid biopsies, there are many challenges associated with them, especially the non-uniformity of coverage. If you're dealing with solid tumors, you're lucky. Liquid biopsies, especially for cell-free tumor DNA data, is quite challenging to handle. Nevertheless, they may help you provide a distinct ordering of mutations, because no matter what, there are ways to add constraints to your topology construction. So I'd like to give one example based on the tool that developed by my lab called SynVict on a number of prostate cancer patients treated in a similar way. The details are not important. So we have data from the time of the treatment, 12 weeks into treatment, and relapse. So there are 3 time points as depicted here. So this is one particular patient, and each position on the horizontal axis denotes a particular position in the androgen receptor gene. So it's a mutational position identified through bulk sequencing data, and we wanted to check how, in time, the mutational prevalence would change in liquid biopsies. And each color depicts a particular time point. Baseline is red. After treatment, 12 weeks into treatment is green, and progression and relapse is blue. As you can see, many of these mutations, and they are all present in the original solid tumor, change their prevalence during treatment. And many of these, in fact, in each loci, you can see that one time point is dominant, meaning the ones that mutations that are significantly prevalent in the solid tumor disappear during treatment. And some may emerge during treatment, but eventually are not found in relapse. There are some other mutations, initially insignificant, that eventually get you. So it's important to identify the temporal order of these events, especially when monitoring recovery. It's difficult to obtain matching solid tumor data and liquid biopsy data too. However, as single-cell sequencing technologies develop, we are given another opportunity to analyze and come up with tumor phylogenes. Since, as you can imagine, if we have the full mutational composition of single cells, provided that we sample at a decent rate, we will be able to have the individual nodes of the phylogenetic tree through which we can obtain the overall evolutionary scenario. I'd like to focus on that bit from this point on. However, I have to say one thing. Even though the single cell data may promise a lot, and you must be hearing about especially single-cell RNA-seq, which is becoming very, very prevalent. Single-cell genome-seq is not very easy to deal with. There are multiple reasons for that. One of them is the doublet issue. How many of you deal with single-cell data and doublets give you nightmares? Yeah, I'm one of them too. These are pairs of cells that have been captured as a single cell. So when you sequence them, you think you're sequencing a single node, but it's actually a mixture again. And that complicates the analysis quite a bit. Even though the doublet rate has been dropping, it's still a major issue. The second issue is subclones with no sample cells, right? If you have this mixture and you sample a small percentage, the blue subpopulation is not represented here, so you're missing an important piece of the puzzle. Non-uniform sampling is another big issue, even though, say, you can see that the pink population, say, is at least as prevalent as the green population. Since you're getting a small subsample, it's very difficult to say how prevalent one subpopulation is against the other. So these are trivial and obvious to notice issues, but bigger issues are related to DNA amplification. Amplification errors giving you false positives are an issue, not as much as unequal amplification, especially allele drop-off, right? So there is this amplification issues creating false negatives, but there are other issues related to the evolution itself. If a certain allele has been lost during the evolution of the tree, then you may be again fooled to believe that the mutation is not harbored by that particular subpopulation, which will again complicate the analysis. So allele drop-off is, for us, the biggest challenge that we'll need to face, which exhibits itself as a false negative in the mutational matrix. So in a typical input of single cell data, basically you'll have the mutations and cells, and each particular mutation observed in a single cell will be depicted by 1 here. Sometimes you cannot really tell. And if it doesn't exist, you just put down a 0. And your goal may be eventually to handle all these issues that give you false positives and false negatives and perform some error correction on this matrix so that you can establish a reliable tumor phylogeny. So as we mentioned, the false negative rate is pretty high, in the order of 15 all the way to 30%, and that is our major concern, especially in comparison to false positive rate, which is relatively small. Doublets, again, complicate the issue depending on the data set. So the 3 inference algorithms that we came up with and some others also came up with typically are based on the following important assumption. We try to establish a tree that satisfies the infinite size assumption. We assume that each mutation occurs once and only once in the entire history of the tumor development, right? So initially, I'll start with this assumption. Maybe I'll say a few words in the end in cases that violate this assumption. So typically, we have a false positive rate and false negative rate that is assumed to be known. We would like to come up with a tree topology and an assignment of each one of these cells to a particular node depicting a clone or subclone, such that the error correction cost is in line with what you expect with respect to false positives and false negatives. You can come up with, again, a combinatorial optimization function to achieve this. Eventually, if this is the true matrix, certain constraints on the mutational distribution across distinct cells may make certain topologies not viable and certain topologies will be viable. And our goal is to figure out which topologies will be viable, especially if we know the number of subclones or the number of clones, the number of nodes in the tree. So one important aspect here is, as I said, when you're dealing with single cell data, it's very difficult to infer the relative prevalence of each one of these populations. And the bulk sequencing data analysis typically provides you this kind of information and close the gap between what you can do with single cell data and what you eventually want to get. So we argue that ideally you'd like to have both bulk and single cell sequencing data, and joint analysis can provide you not only the topology but also the prevalence numbers for each one of these clones or subclones. Obviously, the addition of single cell data constrains the number of topologies quite a bit, so it's a marriage between ideal parties in many ways, especially thinking that bulk sequencing data does not suffer from allele drop-off. So we have a method to jointly analyze single cell and bulk sequencing data in a way that from bulk data we have the variant allele frequencies of each one of the mutations under the assumption that they represent the actual total prevalence of all of the clones and subclones that harbor them, plus the single cell data is just presented to you as a mutational matrix, and you try to integrate them in a meaningful manner. And the end result typically is going to be this mutational tree, which can further be compacted according to your constraints about the number of clones or subclones into a clonal tree. So this kind of a topology where each mutation is represented as a single node is considered to be a mutation tree, and this is the clonal tree. So the end result, the tool that we call B-Site, which has been developed in collaboration with Nico Berenwinkel's group in ETH, achieves this in a combinatorial optimization setting where the goal is to maximize the score that your tree topology achieves based on the false positive and false negative rates in the single cell data, plus the eventually, when you get a clonal tree, the score that you would get for the bulk data. The bulk data gives you, once again, the variant allele frequencies. So in order to maximize the score, we came up with a very simple MCMC procedure, which can be summarized very, very simply. There are many steps that I don't want to get in because you have to really look for a solution in a large space of tree topologies. You'd like to either figure out in each step of the MCMC whether you'd like to alter the tree topology or you'd like to alter the parameters. And if a new set of parameters are proposed with respect to this time false negative rate and false positive rate, based on your observations, I guess these can change, your score will be updated accordingly for a given tree topology that you haven't changed, or if a new topology is proposed, then you have to update the score according to the topology itself, which will have an impact on both the cost of, or the score of the single cell data and the bulk sequencing data. So the tree topology is altered pretty easily. The space is traversed by single edge alterations. So if you can imagine, given a fixed number of nodes in the tree, you're trying to move subtrees from one point to another for traversing the entire tree space. So I'm not going to get into many details. You can find these details in the recon paper. But the end result, I think I'm going to skip some of these. The end result basically accepts the proposed topology and the parameters with a certain probability and continues with the search. So once you obtain a mutation tree, obviously there's a corresponding clonal tree. The clonal tree is obtained basically by grouping nodes that form a path, not necessarily an entire path, but partial paths to fit your constraints about the number of nodes. And once you obtain the clonal tree, then you can, I see. Then you can do a comparison. We have ways to compare mutational trees and clonal trees despite different levels of granularity. And there's a FAB paper that was presented recently in August that deals with similarity between such trees of different granularity under the assumption of infinite sites. You may want to, again, go through that. So I'm not going to get into measures of computing a similarity between trees. But in summary, B-site seems to capture clonal trees from publicly available single cell integrated with bulk sequencing data quite well. There are a few examples that I can go through, but since this is an algorithms workshop, let's skip to the new contribution, which we call physics. Basically, NCMC is not always very satisfying. And either we'd like to, as I have been saying, either we'd like to reduce the problem into a well-known combinatorial optimization problem with well-implemented, fast existing tools. And a few of these tools have been made available for ILP and QIP, as many of you know, integer linear programming and quadratic integer programming. And more recently, we have been interested in the use of weighted max set solvers for combinatorial optimization problems like this. And you can imagine this problem as the construction of a maximum likelihood tree for a given matrix. So let's forget about the bulk sequencing data. Let's focus on the single cell sequencing data. So our goal is to correct or give values to not available entries in the mutation matrix such that a phylogenetic tree can be imposed. So especially if we think that the infinite size assumption work, then we have to come up with a matrix that have to satisfy certain rules. So the rules are very simple. So assume that for now our false negative and false positive rates are fixed, and consider an arbitrary cell Cj assigned to a particular node in a given tree. Then I can easily calculate the probability of observing the cell based on the mutational composition of the clone that is supposed to originate from, right? I can easily calculate the probability. I'll skip the details, but my goal is to come up with a tree topology that obviously maximizes the probability of all cells. So I'm trying to maximize the sum of the log probabilities of each of these cells in the tree space. And for doing that, I should look for trees that satisfy the infinite size assumption that every mutation occurs exactly once in the tree. So that can be summarized as follows. In order to have a single topology that underlies a certain mutational matrix, I have to satisfy the following very, very simple rule. So, just 1 second. Right. I should avoid any cases like this, which can be very, very quickly summarized that having no triplet of cells with 2 mutations such that one cell includes both mutations and the 2 other cells each have a distinct mutation and not the other. A case like that violates the infinite size assumption and should not be allowed. If you can guarantee that there are no triplets of cells and a pair of mutations satisfying this, violating this, then you're guaranteed that there's a perfect phylogeny that can be built by the matrix. And in fact, that tree is unique. So that reduces the problem of dealing with tree topologies to dealing with a very simple matrix. And you can then define an integer linear program, which very simply allow you to search in the space without dealing with all the worries about the tree topology. So that's a nice reduction. And in fact, the ILP looks very, very simple. So let's go through some constraints. In the end, these are very, very simple constraints like these that tell you what kind of values you get in the resulting matrix in comparison to the input matrix. And as long as you don't worry about the number of nodes in the output tree, this works perfectly. Right? It would minimize the total number of error corrections that you'll have to do in a weighted fashion that would guarantee you a perfect phylogeny. Of course, if you add the constraints about the number of nodes and such, and there are cases in which the infinite sites hypothesis is broken, you have to assume a finite sites model, and you may want to minimize the number of such sites in addition to all the things that you have to do to obtain this tree. There's another treatment that you'll have to follow, but all can be formulated as a single quadratic integer program. So I encourage you to consider this. Interesting enough, there's an equivalent weighted max sat formulation that I'm not going to get into that achieves exactly the same goal. And with some of the available tools for solving max sat, you can get reasonable results on typical single cell mutational data in minutes. So again, there are some details that I'm going to skip. The bottom line is, you can solve this relatively well-constrained problem by existing combinatorial optimization packages, but we are thinking of generalizing this to cases where rather than infinite sites, we have a Dolo parsimony model, so we don't have the perfect phylogeny. We are also trying to integrate structure variation, copy number variation, and so on in this whole formulation without assuming that we know them. So copy number variation is essential. You can try to integrate it in a meaningful fashion rather than thinking that it's a part of the input data. We are, like many of you, are trying to use long read or single molecule sequencing technologies. And now our biggest challenge is truly scaling up so that we can build phylogenies for thousands of cells as more and more data become available. And finally, better integration with liquid biopsy sequencing, not as an afterthought, but within the formulation is desperately needed. So with that, I thank my students. My current students are on the left column. My ex-students who have been part of this, such as Iman, Faraz, and Nilgün are in the middle. And finally, my collaborators, Colin Collins and Nico Barenwinkel. And thanks for listening.
Thank you so much for the inspiring speech. I think we can take one question before we move on to our keynote speaker.
So you mentioned it very quickly, but I'm interested in sort of the practical requirements. You said it was like on the order of minutes for how many cells?
Okay. So we are talking about up to 50 cells and dealing with less than 100 mutations.
And then how do you expect that to scale?
So currently, first of all, I have to say that I never have dealt with reliable single cell data with hundreds of cells with many mutations yet. It's all based on simulations. So eventually, especially with MCMC, it's not a big deal. You get what you get. So I don't think that anybody has a much better idea than this yet. But all these are based on branch and bound techniques, especially the MaxSat solvers are all based on branch and bound techniques. And we have to get into how this is solved. So there was a nice proposal by one of the audience members. And we have to probably develop our own techniques based on maybe learning where to look. Because these are general solvers, and eventually I think we have to develop our own algorithms from scratch.
Thank you.
Thank you.